Welcome, everyone, to Slugs and Steins Lectures from UC Santa Cruz. My name is April Yee, and I'm part of the UCSC Alumni Council and one of your volunteer organizers tonight. With me is David Hansen, a previous member of the UCSC Alumni Council, Mike Reapy, a previous member of the Alumni Council and past president, and Diana Hogue and Shana Kent with the Alumni Relations Office. And for those who are new, our Slugs and Steins series engages a UC Santa Cruz faculty member in discussion with you, the local community of the Monterey Bay and Silicon Valley, and our extended community online, with a goal of making us all Renaissance people. And we want it to feel just like you're at UC Santa Cruz sitting in class, but with a drink in your hand and without tests. So before we get started, and since we can't see you, we'd like to know where you're zooming in from and how many people you're watching with. So please fill out the poll that just popped up on your screen, and we'll share the results after we've given everyone a moment to respond. All right, we'll close the poll. And now you should be able to see the results of the poll and who's in the virtual room with you. So most people are watching with just you. Some people are watching with 2, and some are watching with 3 people in the room. And most are watching from Santa Cruz County and the Bay Area and other parts of California, and then other regions in the US. So no one from outside the US or in outer space today. All right. So tonight we'll be tipping our steins with Roxanne Beltran, assistant professor in the ecology and evolutionary biology department at UC Santa Cruz. At UC Santa Cruz, she leads the Beltran Lab, a diverse team of postdocs, graduate students, and 12 undergraduate student researchers. Her lab group uses biologgers to produce interdisciplinary insights. Most of Roxanne's research takes place at Año Nuevo Reserve, where she explores how behavior and physiology interact to drive the ecological and evolutionary patterns we see in nature. Roxanne is a National Geographic Young Explorer, a Beckman Young Investigator, and a Packard Fellow in Science and Engineering. She also authored an award-winning children's book, A Seal Named Patches, which has sold over 15,000 copies worldwide. Finally, Roxanne is passionate about inclusion of marginalized students in scientific research and co-founded UC Santa Cruz's Building a Better Fieldwork Future program, which is working toward making field settings safer, more equitable, and more welcoming for the next generation of field scientists. This evening, Assistant Professor Beltran will discuss open ocean discoveries through partnerships with elephant seals. We'll have a detailed Q&A at the end, but Assistant Professor Beltran will take a few questions during the talk, so don't wait until the last minute. You can type your questions into the Q&A box at any time. And if you see someone else's question that you like, you can upvote it and we'll ask it sooner. In a few days, you'll be able to find this lecture on the UC Santa Cruz Arts and Lectures YouTube channel. We'll post the link in our social media channels and our follow-up emails. Okay. Does everyone have their steins? Great, I've got your slug. Assistant Professor Roxanne Beltran.
Thank you so much, April, for that nice introduction. And thank you all for being here. I know it takes a special type of person to get excited about having a beer and learning about science on a Monday night. So cheers to you. Can you all see my screen okay? April, can you give me a shout if you can see that?
Yep, we can.
Awesome.
Thank you.
Thank you so much. So today I'm going to take you all on a virtual journey up to Año Nuevo and into the dark depths of the North Pacific Ocean, so you can understand how my research team makes open ocean discoveries through partnerships with elephants. So I want to talk to you a bit to start about the open ocean, because it's probably not something you think about on a regular basis, but it's something I think about almost every day. The open ocean is fundamentally really different from everything you and I experience in our day-to-day lives, right? There's not a thing in sight, no physical landmarks, no habitat characteristics, no man-made structures, really nothing on the horizon. And it's so distant from our research stations that it's really hard to study. And as a result, there are a lot of fundamental questions about the marine environment that remain unanswered, like the movements of animals relative to their prey and predators. And this is especially true in 3 dimensions, right? For the deeper parts of the ocean, like the mesopelagic and the bathypelagic zones, which are really devoid of light and limited in oxygen. But the development of instruments attached to animals has really transformed our understanding of the ocean and beyond. So here I'm showing the global migrations of birds and mammals. You can see if you look in the top left corner, the northern elephant seals that are departing from California, and those are the species that my lab group studies. It's really incredible, what these tags can tell us about how nature works. But there's just one problem with this new tool, which is that we have to try to get the instruments back in order to collect the data from them. There are some tags that transmit some data, but to really get the full suite of information about the open ocean, you have to get these tags back. And what that means is you need a really ideal study system to be able to do that. And that's where elephant seals come in. So my team works with what I think of as the perfect research partners, building off research that has been run, for the last 30 years by Dan Costa and his team. So they've led this huge effort to instrument about 40 seals per year at Año Nuevo, and found that they undertake these really extraordinary migrations on their quest to find food and to just stay alive out in the open ocean before they return to Año Nuevo, where they breed and molt. These seals are traveling up to 10,000 kilometers, and throughout that whole trip, they're diving continuously from the surface down to about 500 meters or 700 meters, holding their breath for about 25 minutes on average, followed by just 2 minutes at the ocean surface to catch their breath. And so by instrumenting them with these tracking devices, we can begin to get a better understanding of what's happening in the open ocean. And I don't just mean what the seals are doing, I mean what the physical oceanographic conditions are, how the resource pulses are happening, what the fish abundance and what the whale abundance is. And not just that, but we can put all those pieces together to be able to use these migratory seals as indicators or sentinels of their ocean environments. And it's not just that. It's not just that they randomly sample the environment. These animals are actually really well adapted to be what we call smart ecosystem sensors. So over evolutionary time, they've developed all these incredible adaptations to explore their environment and really hone in on these really productive places. And so they can bring that information right back to us, which is pretty cool and a really remarkable research opportunity. Now, they come back to these beaches along the California coast, and we're particularly lucky that the elephant seals come to shore for a couple months per year at Año Nuevo Reserve, which is what this drone imagery here is showing. You can see those seals that are just hanging out on the beach. You can see that nice equal access boardwalk at the state park and the road that we drive into. And this is just a 30-minute drive north of our university, which is a pretty special opportunity to be able to do the research and to bring students out there as well. And so we can really handle and observe these animals on the beach where they're pretty accessible to us, and they can explore the open ocean. So, like I said, a pretty spectacular partnership. So here's what we're going to do today. I've given you a bit of an introduction. I'm going to give you Elephant Seal Research 101, how we do it and why. And then I'm going to go into a research case study. So I'm going to talk about survival strategies. We're then going to go into some hot off the press research summaries where I talk about some snippets that we've published in the last couple of years. And then I'll give a sneak peek into future research, how to follow it, and then we'll wrap up. All right, so we're going to start this adventure with the seals themselves and how we actually manage to keep track of individual seals. A subset of seals that are born at Año Nuevo every year are inducted into our research program as pups. And what we do is we really carefully capture them in this red canvas bag after they've weaned from their mom's milk. And then we weigh them to see how successful their moms were in the previous foraging trip to sea. Then we give each seal a flipper tag that has an alphanumeric code that becomes its identifier, sort of like its social security number for the rest of its life. And then we walk around the beach and we observe the seals year after year in order to see how they survive and how they reproduce. Now, you can imagine standing in a sea of elephant seals, especially during the breeding season, it's pretty hard to see those little plastic tags. So although those plastic tags stay on the seals for their whole lives, we actually do one more thing, which is that we add a dye mark to the sides of the seals every year with a much bigger number that looks like this so that we can see them a lot easier. And as you can see, they don't react really strongly to marking. We do this with Clairol hair dye in case you're curious, but I would not try this at home. So then we walk around the beach and we're able to see those individual seals, like this one here marked 4235. She happens to be our favorite of all time. We can watch them interact with their pups, so we can tell if they've been reproductively successful in a year. We can see how long the pups are nursing, how big they get, who the pups are, so we can start to build pedigrees and figure out which ones are related to each other. And we can just walk around the beach like this, observing them and seeing the same individuals year after year. And that allows us to get this incredibly precise data about all sorts of things, but most importantly, their demography. And I'll show you what that looks like. So here's an example of a summarized data set from this particular seal, B95. She was first tagged as a pup in 2015. She's now about 8 years old. She's been seen every single year throughout her life, over 200 times total. And those sightings give us really useful information about where she was, whether she had a pup, whether she was replacing her fur during the annual molt. And then we can make any comments like, oh, she had a shark bite, or she was, holding her flipper like it was hurting or something like that. And of course, it's not just one animal we have data for. Over 6 decades in this program that was really started by Bernie LaBeouf in the 1960s and continued on by Dan Costa in the 1990s until now. There's 6 decades of data, 360,000 sightings of 50,000 seals. So it's this really amazing, amazing opportunity to understand the life histories of the animals. And not just that, but also to selectively instrument seals with special characteristics to understand the open ocean. And in addition to instrumenting them, we can also figure out how fat they are. So here's an example of what a seal looks like before a foraging trip when she departs. And when she comes back, she's nice and fat. And we can weigh the seals before and after to be able to measure their foraging success at sea, or the number of kilograms that they gained during that foraging trip. So it all sounds pretty easy, right? You go to the beach, you watch the seals. I mean, it's pretty dream life, and it is. And it also takes a lot of time from a lot of people. And so I want to take this opportunity to acknowledge that my lab group is absolutely amazing, and they work their butts off to get this work done. And it's not just my lab group now, but it's the last 6 decades of people who've been collecting these hundreds of thousands of observations and entering them into the computer every single day. From taking, things like first aid training to make sure that we're up to speed on best practices, to boating to the island when the seals decide not to cooperate and they hang out there instead, to chasing seals to different colonies like Point Reyes and San Simeon, to doing things like recovering instruments in the rain. These guys are always really good sports, and it's always really special to be able to have those long seasons pay off with them. So I just want to give a little cheers to my field crew and say thank you to them for making this possible, because it really, really takes thousands of hours every year to make this possible. And I wouldn't be able to present this research without them. So on to a research case study on survival strategies. I'm going to go pretty deep into this research case study because I want you to understand the sort of scale of research that we're able to do with this incredible data set. So for this example, we're going to answer the question, what are the consequences of behavioral strategies, so what seals are doing, to foraging success and lifetime fitness? This work was co-led by my postdoc, Keith Hernandez, and it was just recently on the cover of Ecology Letters, one of our favorite journals. So we're really excited about this work. And I want to introduce you to the question, which is a really simple question, which is, how long do mammals live? That may sound really ridiculous and simple to you, but it's a really hard question in biology. So let's take elephant seals. The oldest elephant seal ever was 23 years old, that we know of. But on average, elephant seals live to be only 1.4 years old. And it's the same story with a lot of species. For example, here's a little brown bat. They can live to be 34 years old, but on average, they live to be only 2.2 years old. And this is in pretty stark contrast, right, to things like domesticated dogs and humans that often live a really long proportion of their maximum lifespan. And the reason for that, of course, is that wild animals have selection pressures. They have predators roaming around trying to eat them. They can, have to deal with starvation if they don't find enough food. And so one of the really important questions in our field is, what can animals do to stay alive? What strategies are they using in order to avoid predation or avoid starvation? And we can get some hints if we look at the variation in traits across individual animals, right? So if you think about like body size and anatomical feature, there's a ton of variation, even within a single sex and age class. Same thing with physiology, tons of variation in performance. There's lots of variation in habitat selection across individuals, in the timing of activities, and also in things like reproductive strategies. And so if we take all of this variation across individuals and we pair it with other types of variation, we can begin to understand mechanisms that are driving those patterns. So let me give you an example. In the elephant seals, we see extreme variation in demography, so in survival and reproductive success across individuals. I want to give you an example of what some data looks like. So this is data from one seal. Her flipper tags is J493. And as you can see, she was first seen at 3 years old and seen all the way until she was 20 years old. Those are the columns of that table. And then in each of these cells, you can see whether she was missing, that's a 0, whether she skipped breeding, which is a 1, that means she was like on the beach but didn't produce a pup, or whether she produced a pup, which is a P. So this is what we call an encounter history. This is a history of that entire animal for her entire life every single year. And you can see it's a mix, right? Sometimes she's missing, sometimes she's there with a pup. And that's just one seal. For that one seal, we can calculate those lifetime metrics. So here she lived to be 20, and her lifetime reproductive success, or the number of pups she produced in her lifetime, was 5, right? So we can do that for that one seal. We can do it for a second seal and a third seal. And I'll give you a spoiler alert for these particular seals. You can see all of them produced 5 lifetime pups, but they got to that lifetime reproductive success in really different ways, with some seals not living very long, but producing pups every single year of their lifespan, and some seals only producing 5 pups, despite the fact that they lived a really long time. And if tables don't work for you, I'll show this in graphical form. Here are those 3 seals in this 2-dimensional space. On the x-axis here is lifespan and number of years, and the y-axis is the lifetime reproductive success, the number of pups. That white line, falling on that white line, means that you produce a pup every year of your reproductive lifespan. And you can see where these 3 seals lie, hugely variable in where they lay on this graph. And we have these data for almost 1,900 animals now, lifetime fitness data from 1,900 wild animals, to show us the incredible variation in how these seals make a living, how they reproduce, and how they survive. And we can begin to understand that variation by using the variation in behavior and foraging success to try to explain it. And so that's what we've done here. Here on the x-axis, we have mass gain during a foraging trip. So did the seal gain 100 kilograms or 300 kilograms during this 7-month-long foraging migration? And then on the y-axis is the annual probability of survival. So of all the animals that were around, what proportion of them survived? And what you can see is positive trends for all ages. So when seals gain more mass during their foraging trip, they're more likely to survive. Now, if I show you the same plot, except instead of on the left y-axis having survival, I want to show you reproduction, you can see that seals that gained more mass, so the right-shifted ones on the plot, were much more likely to successfully produce pups. And you can see that relationship is super steep. We call that a physiological tipping point. That basically means that a small increase in the amount of mass gain during the foraging trip can make for a huge increase in the proportion of reproduction. And likewise, a small change in the ocean that results in changed foraging success for the seals could have potentially really catastrophic consequences for their populations. So by combining all these things, we're able to better understand how things like foraging success in the ocean influence reproduction and survival. And I'm not going to go into details, but we were able to actually calculate which behavioral strategies were more successful strategies. And those were seals that dove deeper, that foraged further from the coast, and that had more fish in their diet. So we can really begin to understand what the seals are doing, how successful they are, and the consequences that those decisions have on their success. Now, why does that matter? Well, zooming out, this is important because where animals go and what they do and how successful they are has really important implications for their ecosystems and for the ecosystem services that they do. So animals are doing all these things all the time, and those have important consequences, right? Like cycling nutrients and depleting prey and offloading contaminants and facilitating gene flow and transmitting pathogens. And so it's really important to be able to study these behaviors and to figure out the ecological consequences of those behaviors. And that's something that we're working on now. Okay, so that was your little research case study. And I want to shift to hot off the press research summaries. These are, 1 or 2 slides per research study that we've either published or are publishing right now. So I want to start with the very basics, which is a question I get almost every time I talk to docents or to visitors at State Park, which is, when do the seals do X? When are the seals breeding, or when are the seals molting? And it always makes me laugh because it seems like a really simple question, right? But like I said, there's a lot of individual variation across animals. And so we've done a lot of work to just try to figure out this annual cycle. So I'll orient you to it. The outsidemost circle is a calendar, January all the way through December. The color dots in the middle are what we call life history events. So these are things like arriving from a foraging trip, giving birth, weaning a pup, departing for a trip to sea, and then arriving, starting the molt, ending the molt, and then departing. That's a whole year in the life of a seal. And the arrows just outside of that are just to kind of help orient you. It's either, the breeding season, which is here, or it's the molting season here, or the seals are at sea during either this short foraging trip between breeding and molting, or this long foraging trip between molting and breeding. So if you have an astute eye, you'll know we're about right here. We're just before the breeding season. So we're waiting for the incoming chaos of the seals to all show up on the beach at the same time. So one of the first questions we asked was, okay, if they have these 7-month foraging trips, then when do they arrive on the beach and when do they give birth? Basically, what's the difference between these two dots? And we found that seals basically come onto land after 7 months of foraging with only 5 days between arriving and giving birth. So they have this incredibly precise navigation ability that they're able to use to get to the breeding beach right on time. Another question we get a lot is, when does the molt happen? So for those of you with a dog or a cat, you're probably familiar with shedding, like fur replacement. Like I have fur all over me for my dog and cat right now. And seals do a really similar thing, slightly different because they actually molt some of their skin along with their fur. But you can really easily see whether seals are 5% molted, so you can see that new silvery coat kind of popping through on that bottom picture, or whether seals are 50% molted, or whether they're 95% molted. And so we walk around the beach, and on our data books, we record the seals and their molt statuses. And what we can do with those data is we can create this basically progression for each seal. So here on the x-axis, I have date. On the y-axis, I have the percent of the coat that's molted. And you can see for a single seal, this is a seal in 2022, that she went from 0% molted to 25% to 75%, all the way to 100%. And we can fit this model to be able to pull out things like arrival date. When did we first see the seal? Then the molt start date, when that curve starts to get really sharp. The molt end date, when it starts to get really flat again. And then the departure date, the last time the seal is seen. And this is for one seal, but if we take those 4 data points and we plot them for all 484 seals that we have data for, you can see that there's an incredible amount of asynchrony across individuals. So these are organized by molt start date, but you can see that there's a range of 80 days across which seals arrive, which is pretty crazy, right? So when people ask me, when does the molt happen? I'm always like, what? Which sex, which age class, which individual? There's a lot of questions, but my question is, why is there so much asynchrony across individuals? Why are individuals so different? And I don't know the answer, but what I do know is that it has really important ecosystem consequences. And it's really important as well to know how long the actual molt is. So we found that the molt only lasts 7 days. So these animals have a range in the start date of 80 days for something that only takes a week. That means there's 10 times as much variation as the duration of the thing itself. And that's something that one of my undergraduate students, Raquel Lozano, published in her undergraduate thesis, this whole molt story, and how interesting it is that it's so variable across individuals. And since she graduated, I've been trying to dig into this a little bit further to really understand the variation in the population. And this is a plot I put together that shows the variation across the life history events. So how does the variation in breeding, which are the blue and purple dots, compare to the variation in the molting season? And what you should be able to see is those super wide error bars, the super wide confidence intervals in the molt is really, really different than the breeding season, right? The breeding season is much more synchronous than the molt. And if you think about the function of the breeding season, you probably can guess why this is over evolutionary time, right? the males and females have to be there at the same time or reproduction doesn't work very well. And so they've evolved this really incredible ability to be able to come to the beach at the same time to be able to facilitate breeding synchrony. But the question then becomes, how do they actually facilitate that breeding synchrony? And the answer lies in another undergraduate senior thesis, this time by Alex Yuen, who asked a really simple question of when do seals turn around? So he went and quantitatively picked all of the points in the seal tracks when each seal began to return home. And what he found was that the seals that turn around when they're further away allocate extra time to get back if they have further to travel. So this basically tells us that seals not only know their distance to the breeding beach, but also know how long they have to get back before they're late for the breeding season. This is called a map sense in our field. It's like this gold standard of navigation, like, oh, wow, maybe animals have a map sense. It's like you turning on your GPS to go somewhere, and your GPS knows if you're further away, it tells you to leave a little bit earlier. And it looks like these elephant seals have the same thing, which is pretty cool and helps to explain not only their amazing navigational feats, but also how they're able to breed synchronously every year, which is something that we're really excited about. Okay, so with that, I want to take a little break, make a toast to elephant seals and their incredible navigation abilities, and I guess open it up for a couple questions. I'm going to need some help with that. Then we'll have a little Q&A poll to sort of break this up, and then I'll just do maybe like 10 more slides, and then we'll be done with the science for the night.
Sounds good, Roxanne. Yes, Van, we have a number of questions. And by the way, just to remind you, please put your questions down in the Q&A box and upvote those you like, and those will be asked first. Van is first out. He wants to know about the pups and the moms. How do you identify who belongs to whom?
That's a really great question, and it's a very complicated answer. It takes a lot of really careful field work. So I'll give you one example. Let's say there's a mom and she has a flipper tag and a mark. So we see her on the breeding beach. We watch her get back. She arrives. 5 days later, she gives birth. Then we watch that pup, and we watch that pup get bigger and bigger and bigger every day until 20 days after that pup's birth, it gets to be a balloon from all that milk. It gets nice and fat. And at that point, the pup is big enough for us to be able to add a dye mark. So you saw that video of how we dye mark the adult females with a dark dye color. For the pups, we use bleach because they have really dark coats. And so we're able to give that pup a bleach mark that contains a subset of the mom's identification numbers shuffled around in a specific way to mark the pup. So then the mom has a mark, the pup has a mark, then we see them together for a few more days until the pup weans. And then the pup's all alone, safely separated from the mom, and then we can add the pup's flipper tag. Once the pup gets flipper tagged, then we know it's like for sure that pup is going to be able to be individually identified for the rest of its life. So it's kind of a multi-step process because the moms are really big and they have very large teeth, and we don't want to try to flipper tag the pups when they're still nursing from their moms. But after that, then we know the association between the mom and the pup, and the database keeps track of that for us. So for example, we have a 7th generation pup that was born last year. So we know that like 7 generations of moms that came before it, which is a pretty cool thing to be able to do. And the data just get more and more valuable every single year.
All right, thank you. Barrett says males travel less distance from shore and dive less deep than females, so they are poised to have less survival success every year, as we know. Why do they put themselves at this disadvantage? Is it because they need to access more prey over a shorter time period and therefore make this trade-off?
That is a fantastic question, and it is the topic of one of the biggest grants that we have right now, looking at risk-reward trade-offs in the open ocean. So our hypothesis from decades of research trying to piece together little pieces of the puzzle is that, yes, males die more often at sea, and they go to these places that are hypothesized to have more killer whale predators. And we think that the reason that they take that high risk is to get high reward because there's more prey in those areas. Why do they need more prey? They need more prey because they're huge. These seals are super sexually sized dimorphic. So the males are 3, 4, sometimes 5 times larger than the females. And that's part of their breeding strategy. The male's nice and big. They can basically earn their access to the females to be able to breed with them. So the bigger the male, the more successful they are. How do you get big? You eat lots of food. So we think that it has to do with basically sexual selection pulling male body size larger, and then the consequence of that being natural selection pulling it smaller because of the predation risk of the areas with lots of food. But it's a really complicated question, and actually teasing apart those differences with data is actually a really challenging problem. So we're doing our best.
One more question, perhaps, before we move on, and that is: This is from Mary. Do adult males molt their chest shield skin?
Yeah, that's a really good question. The short answer is the molt gets a little funkier on the chest shield. The chest shield is kind of a different type of skin than normal elephant seal skin. And so they certainly molt around it, and the skin sort of molts off the extra layer. But I would say the chest shield is more like scar tissue than it is actual skin. And so whether that counts as molting, Mary, I'll let you decide, but it's probably some halfway answer between yes and no.
You want to do one more question, or?
Let's do it. We're on a roll.
So Jason asked, can you detail a few policy changes you'd like to see from the federal or state level to help elephant seals? Extended protections for marine areas, action on climate change, etc.
That's such a good question, Jason. Oh my gosh, I could talk about policy for like 10 hours, but the short answer is it's policy that helped save the species in the first place. These seals were nearly hunted to extinction, down to about 20 animals, and the Mexican government basically protected them and said no more. And there were a small number of individuals that managed to survive on an island in Mexico, and then slowly but surely over a bunch of generations, the seals have grown back to 220,000 and sort of recolonized the areas that they didn't used to be in. So they're what we use as sort of a conservation success story of the power of policy. In terms of actual policy changes that I'd like to see, I mean, what is the biggest threat to elephant seals? In my personal opinion, I'm a demographer, so a lot of different people would probably answer a different way, but I know that oceanographic conditions like El Nino and like, heat waves and things that are caused by man-made sources cause a lot of cascading impacts on the ecosystems beneath the elephant seals, all the way from the phytoplankton to the fish to the seals. And we know that seals really struggle a lot with years that are bad. When oceanographic conditions are bad, they have longer trips, they go to different places, they're less successful at breeding, their pups are less good, and the pups go on to reproduce less. And so I think anything that sort of helps prevent sort of catastrophic shifts in the baseline oceanographic conditions from climate change would be really impactful in terms of helping the seals do what they're good at, which is going and finding food and avoiding predators in the open ocean.
Great, thank you.
Do we have a poll now?
All right, so this poll is going to lead us into our next research question. So I'm curious what you guys think. We've talked a lot about how elephant seals are amazing athletes. They can, dive really deep and they can go really far, but all animals have to start young, right? So how long can 1-year-old elephant seals hold their breath? Go ahead and take your guess, and then we'll chat about the most common answers. All right, so it looks like most folks were right. 39% of you guessed 30 minutes. So yes, 1-year-old elephant seals can hold their breath for 30 minutes, which probably seems crazy, but I'll tell you about how we learned that. 38% of you said 20 minutes, so that was a really good guess as well. And then it looks like about 20% of you said either 5 minutes or 10 minutes. So awesome guesses. All right, back to the slideshow. So how do we figure out how long young of the year elephant seals can hold their breaths? Well, we had a postdoc in my lab, Geoffrey Juma, who's now on to industry in Europe, doing some really cool data science. And he was interested in development. And he asked me, these elephant seals are really naive when they go in the ocean. Can we instrument them and understand how their development works? And so we put these satellite tags on the heads of some elephant seals. And this is an example of one set of data. So on the x-axis here is date. This is a 215-day migration from one juvenile elephant seal. And you can see that aerobic dive limit, the behavioral aerobic dive limit, basically how long the seals can hold their breath in minutes throughout that whole trip. That's on the y-axis. And so what I want you to see is there's an incredible amount of diving development in that first year. This particular young of the year tripled her diving capacities in that first year. And in case you're paying a lot of attention, you remember that video I showed you at the beginning, the BBC video that had that seal 4235 that I said was our favorite. This is actually her 9th pup that we were able to instrument. And so we were really excited to see that, that mom has really amazing diving abilities. And this seal in particular had really amazing diving abilities as well. So one of the questions we have is, is it heritable? Are there heritable abilities, diving abilities that are passed from mom to pup? In addition to the sort of diving capabilities at the baseline level, we also want to know how oceanography is impacting the seals. And so in this particular research vignette, we wanted to ask whether oceanographic conditions drive seal reproductive success. And so this was actually a class project. I teach a field methods class called 128L, and we have 14 students, and they tackle some sort of research question together. They work as a team. And this year they said, we want to figure out what proportion of pups in each cohort since the 1980s live long enough to produce pups of their own. We call that recruitment success in our field. And here's the answer to that question. You should notice that there's a huge amount of variation in the success of each cohort. And it's not just that there's a lot of variation in success, is that there's these sort of cycles, right? You can see peaks every 10 years or so where the seals are doing really, really well. And so their question was, well, what's driving these population cycles? Could it be oceanography? Is it possible that the oceanographic conditions are driving these pulses in elephant seals? And so they overlaid the oceanographic data and saw that, in fact, the good years, so here shown as the more yellow colors, this is the North Pacific Gyre Oscillation Index, basically an index of how healthy the ocean is in a good year. You can see that those yellow years are when the seals are doing really well. And then when the oscillation index is really low, when it's those purples and blues, you can see the seals are not doing as well. So there's clearly a lot of correlations between how successful the seals are and how good the ocean's doing. And we'll come back to that later when I tell you that this year is a really strong El Nino year, and it's going to be really interesting to see how the seals are able to sort of respond to those oceanographic conditions. But it's not just the prey. We call this bottom up in ecology. Like, it's not just the oceanography and the fish that are driving the seal population dynamics. It's also the predators. And so in the next research project I'm going to show you, we basically tried to figure out if seals are showing signs of predator avoidance during their trips to sea. And I'm going to play a video that sort of summarizes the research findings because I couldn't possibly explain it as beautifully as this animation by Jesse Kendallbar. I just love that example because it sort of summarizes a lot of the work that we're doing in trying to use biologgers to understand elephant seals. And I want to talk about one more research snippet, which is on more of the sort of broad science that my lab group is doing. This talk focused on elephant seals, so I didn't get a chance to talk about a lot of the important work that folks in my lab are doing to try to make the field a better place. But one example of some of the sort of broad scale work that we're doing is a manuscript that my PhD student, Allison Payne, just finished about the ethics of handling wildlife in partnering with animals to understand them and the ecosystems. And the idea is that for animals that walk or swim or fly, there's different impacts that these biologgers can have. And we want to make sure we're minimizing or completely getting rid of any impacts that we're having. Because if we're partnering with the animals to understand their behavior, we want to make sure it's their natural behavior. If we're partnering with the animals to understand their ecology, we want to make sure that the instruments we're using aren't messing with their predator-prey interactions, for example. And so Allison and her undergraduate student mentee, Connor, and Jesse Kendallbar, who illustrated these figures, did this beautiful review about the potential impacts of these instruments. Elephant seals are, of course, one of the biggest animals we can instrument, and so the impacts are really, really negligible, which we're really happy about. But that's not the case with smaller animals and animals that fly and therefore have to carry instruments in the air, or animals that live in certain habitats or have certain behaviors. And so Allison wrote this really beautiful review about which studies have found impacts, which haven't found impacts, and on which taxa and species and habitats those are in, so that we can better inform the research that we're doing and sort of take our research and the rest of the biologging community and say, hey, how can we be better partnering with animals to protect them so that we can understand them in their natural states? And I'm really, really proud of the work that these guys have been doing on that. Okay, so we've made it through the research case study. We've made it through the hot off the press research summaries. I want to give you a sneak peek into future research and how you can follow it. One of the best ways is by going to our lab website, beltranlab.ucsc.edu. Currently, we have 2 sets of animals that are instrumented, 4 to 9-year-old females. There's about 12 of those still online. You can see that's the top plot. They should be returning in the next 2 months or so for the breeding season. And then this bottom plot, which has 0 to 3-year-old juvenile males and females that are part of that project I talked about, looking at risk-reward trade-offs and how males and females navigate space differently based on their needs. You can also go to the Facebook page, facebook.com/OnUnuevo-research, to get more broad updates about some of the bird and sea lion and seal work that's happening with other groups at the reserve. And then I couldn't bring my lab with me because there's a lot of them and we only had 45 minutes. And so I couldn't represent all the research that our group is doing, but I asked each of them to put together about 20 seconds of video introducing themselves and the work that they're doing, so I could give you all a sort of broad sense of some of the other questions that people in my lab group are asking. So I'm going to play for you now a 3-minute video of the rest of the lab sort of introducing themselves.
Hi, my name is Allison Payne, and I'm a PhD student in the Beltran lab. My research focuses on a novel acoustic recorder that can be attached to the back of elephant seals so that we can listen in on the deep ocean, including some rare kinds of whales like beaked whales.
Hi, my name is Dee, and I'm a PhD student working on the NSF-funded Rules of Death Across Ontogeny project. As part of this project, I'm doing the physiology component, looking at how juvenile elephant seals develop in the early stages of life. I'm accomplishing this by a combination of blood samples, biological measurements, and tissue collection.
Hi, my name is Lina Arcila Hernandez. I'm a postdoctoral researcher in the Beltran lab. I'm also an evidence-based education researcher, and I'm really interested in understanding and developing best teaching strategies that can create inclusive field-based educational experiences.
Hi, my name is Natalie Storm, and I'm the lab technician in the Beltran lab. My job largely consists of supporting the long-term northern elephant seal mark-recapture program at OnUnuevo. I spend most of my time out in the field with undergraduate and graduate students conducting flipper tag and markery sites, as well as weighing and tagging end-of-the-year seals. In addition to supporting ongoing research efforts, I am largely interested in reproductive trade-offs in adult female seals and how decisions such as skip breeding and altered behavior affect their long-term reproductive success.
Hi, I'm Connor. I'm a program assistant and former undergraduate research assistant in the Beltran lab. My research focuses on the predator avoidance strategies of female elephant seals and the ethics and best practices of biologging.
Hello, My name is Cara Monroe. I'm a program assistant in the Beltran lab. I'm interested in the monitoring and management of wild marine species, and my research spans from modeling the impacts of killer whale consumption on Chinook salmon to understanding how senescence in northern elephant seals affects maternal breeding.
Hello, my name is Bella Garfield, and I'm an undergraduate assistant in the Beltran lab. My research is looking into habitat switching by elephant seals, specifically pertaining to site fidelity and how female elephant seals use the beaches at Año Nuevo State Park during the breeding season. I also help out with some drone projects, as well as helping to maintain our large resite seal database.
Hi, my name is Astarte Brown, and I'm a current undergraduate student researcher in the Beltran lab. My primary role in the lab is to assist in the long-term population monitoring efforts of the elephant seals. But in addition, I've also been contributing to a research project studying adult female elephant seals and their diving behavior in relation to predator presence.
Hi, I'm Esme Singer. I'm an undergraduate field assistant in the Beltran lab, and my senior thesis research is on seal whisker isotopes and what they can tell us about where the seals are foraging in terms of trophic levels and latitudes. And my research is particularly focused on how these isotope ratios vary in La Nina versus El Nino years.
Hi, my name is Maddie Stewart. I'm a third-year undergrad in the Beltran lab. My senior thesis includes comparing the head, teeth, and blipper sizes of juvenile northern elephant seals in order to understand juvenile development and sexual size dimorphism. My other research interests include the genetics and behavioral ecology of large marine mammals.
Hi there, my name is Kelly Young, and I'm a third-year undergraduate student here in Roxanne Beltran's lab at UCSC. This year, my senior thesis work is about finding new and safer ways to measure elephant seals. And one exciting way I hope to do this is by using drones. I'm also an undergraduate research assistant, which means I get to go out each week to our colony of elephant seals at Año Nuevo Reserve and help to recite seals or help out on procedures, which is really, really fun.
Hello, my name is Rye. I'm the Beltran Lab mascot. My research question is, where does everyone go each day?
My husband Patrick stuck that last one in there, but in all seriousness, my team is incredible, and like I said, it takes a ton of work to get this done, and I'm so lucky to be surrounded by such bright, inspired, exciting people. Like I said, I didn't get a chance to talk about a lot of the work everyone's doing, and I didn't get a chance to talk a lot about the sort of equity and inclusion efforts that our lab is doing, but I just want to say that in addition to paying all of our field assistants as undergrads, which has been really, really amazing in the last couple of years, we've also tried really hard to purchase shared gear because that removes barriers to participation for low-income students. So we bought a truck. That was a pretty big deal. You can't really see it, but there's a truck behind all those guys. And we were able to buy boots to be able to outfit everyone as loaners so that people don't have to wear their shoes from class to the field and seal poop and, disintegrating kelp and stuff like that. But one thing we don't have, which is warm jackets. And so as you can imagine, working in the winter at Año Nuevo can be really cold and really wet. And in this year in particular, it's supposed to be a super El Nino. It's supposed to be one of the wettest years we've had in a really long time. And so I was told by the organizers of this that I was allowed to ask for a donation at the end. And so I just want to ask if you are warm and cozy in your house right now, just imagine the field assistants out in the field in the rain, trying to collect these data to keep the long-term data set going, and consider donating a couple bucks if you can to help us buy some warm jackets so that they can be nice and warm this winter. So with that, I will just thank the funders of our lab. This research is really exciting and it's really expensive. And so we're really lucky to have all these people sort of backing us and making sure that we can get the work done. To our science communication partners, to the docents at Año Nuevo State Park, at the Friends of the Elephant Seal, and at the Seymour Center. I know a lot of you are on this call. Thank you so much for the work that you're doing. We really couldn't make the impact that we're having without you all. And to the department community, to my personal mentors who've really inspired me to do this work and who trusted me as a paid undergrad student 10 years ago, which allowed me to get to where I am today. And then finally, just a shameless plug that if you are interested in hearing more research or watching me experience Galapagos sea lions for the first time, which is going to be amazing, I'm leading a trip to the Galapagos in June of 2024, along with the Reserve Director of Año Nuevo, Patrick Robinson. We are going to go for 8 days on this inspired expedition. We are so excited. It's going to be an amazing boat trip, and we would love for you to join us. So if you're interested in that sort of thing, go ahead and navigate to this website, shoot me an email, send an email to the travel folks at UCSC, and we'll try to fill up the trip. It's coming up soon. So with that, Thank you very much for your time. Cheers to science and to Steins. And I guess I'll take any questions. Thanks so much.
Well, that was marvelous. Thank you so much. Of course, there are questions galore. We'll start with Stacy. Why Año Nuevo? Is there a reason Año Nuevo is their home base? Are there other areas that host lots of elephant seals?
Yeah, that's a super good question. And the short answer is I can guess all I want. And the best reason I can find that the seals like Año Nuevo is that it's, as you can see, pretty flat, pretty nice, has lots of other seals in it. It's also adjacent to an island. So you can see the island in the background here. When seals were being hunted by humans, they inhabited mostly or exclusively islands. There were also bears back in the day on the mainland. And so a lot of the early colonies were on islands and they sort of spilled over to mainlands. And so it makes a lot of sense for them to come to places like Año Nuevo, where the island and the mainland are just a kilometer away. As the seals sort of re-expanded and recolonized area after they were, basically rescued in Mexico, they started going north and north and north and north up the coast. And so there's something like, I actually don't know, maybe 18 or so 15 breeding colonies along the California coast. Someone probably knows. And there are huge colonies like on the Channel Islands and at Piedras Blancas, where there's an amazing docent program, Friends of the Elephant Seal. Año Nuevo is actually a pretty small colony in the grand scheme. It's something like 10,000 animals, throughout the year, whereas some of the other colonies are 10 times that. And then there's some really small breeding colonies north. It seems like, the seals are sort of shifting northward. And I don't think we really know if that's because of climate change or if it's because the population numbers are increasing and they just need somewhere to go. But Año Nuevo has been a pretty consistent place for them to haul out, and they seem to be doing pretty well there. So we're pretty excited.
Awesome. The next question is from Stuart. What are your selection criteria for which elephant seals you pick to tag with the head data collection device? And how are your tags attached to the seal's head? Do they fall off and get lost?
Hopefully not, because each instrument is something like $5,000 or $6,000. So we try to attach them pretty firmly. We do that by sedating the animals really carefully with a trained team. And so we sedate the seals on the beach with a team of usually 4 or 5 other people. We carefully, when the animal's under anesthesia, then glue the instrument to the seal's head, to the fur. And as lots of people have noticed in the Q&A, during the molt, the fur falls off. And so we usually try to get the instrument back before that. When the seals return to Año Nuevo, they get to the beach and then we sedate them for a second time to remove the instrument. That's what we call our recovery procedure. And then the seals wake up once the ketamine wears off and they just go on their merry way. It's a really safe way of handling them, both for them and for the researchers. And so we prefer to do it that way. In terms of which animals we select, it's a really great question. It depends on the research project. So for the long-term biologging monitoring, it's always been adult females that have produced at least 1 pup at Año Nuevo, so we know they're going to come back. And that's a way that we get those really expensive instruments back. But for example, for the juvenile project I told you about with the risk-reward trade-offs, those are 0 to 3-year-old males and females. And so that's just a fundamentally different subset of animals. So my graduate student Salma has this code she runs on her computer to basically make us a list every day that she prints for us and says, these are our candidates for today. And then we walk around the beach trying to find one of the 20 animals on the list that meets our exact specifications. And as soon as one of us finds that seal, we radio to the others, we grab the gear, go to the seal, sedate the seal, and then check that animal off our list. And so that's how we do different research projects. But the beauty of the seals is that we can ask different research questions with different age and sex classes. So it really depends on the day and the time of year.
Hey, by the way, just to remind you, please add your questions and please do the upvoting. Help us pick which question to ask. Jessica asks, do you lose identification marks when the seals molt?
We do. It's so sad. Every year we have to remark the same seals all over again. And so it's a lot of work and we go through a lot of hair dye at $8 a bottle. But it's totally worth it because it's a lot less invasive than some of the other methods that are used by some research groups. Like in the, way past history, people would actually brand like hot brand iron animals because those scars stay for the whole seal's life. And, we don't want to do that sort of thing. And so marking is a lot of work, but it's a lot better than the alternatives for a sort of like animal best practices standpoint. So we're happy to do it. And the seals don't seem to mind too much.
Got it. Stuart asks, do the seals return to other beaches in other years, or do they always return to the same beach?
It's a great question. And the answer is it depends a lot on the age and sex class of the animals. So I told you the long-term program mostly tags adult females, and that's for a very specific reason. And the reason is that once females reach sexual maturity, they have super high site fidelity. Like they come back to their natal beach year after year after year with a lot of confidence. And this juvenile project we just started is kind of the first time that like a major research effort has been done on juveniles. And so we weren't really sure like how much the juveniles, what we call prospect to other colonies. Like we have a whole system along the coast of researchers that sort of coordinate to put out specific flipper tag colors. So if you were paying attention, you saw that Año Nuevo has green flipper tags. The Piedras Blancas colony has white flipper tags. Point Reyes has pink flipper tags. And so we can tell just based on the flipper tag color where the seal was born. And you can tell during certain times of the year, like the juvenile haul out period, which is kind of happening right now. It's like Skittles out there. There's a pink tag, a green tag, a white tag, a yellow tag, a red tag. They're from all over the place. But during the breeding season, almost all of the tags that we see are green. And so that gives you a sense for the fact that the juveniles are much less site fidel than the adult females, and means that if we instrument juveniles, they're very likely to show up at other colonies. And so as my team knows, we've had a pretty exciting couple years of recovering instruments at all sorts of places, Piedras Blancas down south and Point Reyes up north, and seals that have gone to other places that we couldn't really access. And that's because the juveniles are just trying to figure out where they want to breed for the first time. Once they breed, they pretty much go to that same place. So it's been an exciting couple of years, but we never say no to an opportunity to visit the Piedras Blancas colony and see the docents down there, or to go to Point Reyes and see the Park Service staff up there. So we don't complain too much, and it's fun to be in the car.
Joanne asks, at what age do male and females' feeding strategies start to differ? Where do juveniles typically forage?
That's literally the main question of the really big grant that we have. So I told you it's about risk reward trade-offs, and that's true. And the question is about when the males and females diverge and how they're using those risk reward landscapes. And so that's why we're instrumenting both females and males, is to figure out when they start doing different things. I'm not going to ruin Salman Z's entire dissertations by answering that question for you, but I will say that there's a very obvious difference in when the seals go to the pelagic areas like the females do as adults, or to the coastal areas like the males do as adults. And the females change depending on whether they give birth or not. So the seals can start reproducing at age 3 or age 4 or age 5. And we see 3-year-old females returning, some with pups and some without pups. Like some have decided to start reproducing and some have not. And those two types of females do really different things. They have different timing. They have different locations that they're foraging in. So it seems like probably right before sexual maturity is when they start to diverge, but it's not a specific age. It also has to do with how reproductively successful those seals are. So hopefully we'll have some published research on that in the next few years. It's a great question.
Yeah, this next question from Michael is somewhat related. It says, any insight into when a female elephant seal becomes pregnant for the first time? Is it strictly during breeding season, or can it occur in the late fall?
Yeah, that's a really great question as well. And the short answer is we're still learning a lot about how seals get pregnant. And that sounds like a really weird thing to say, but seals have what's called diapause. So they have this little break between when they're bred by the males and when that blastocyst sort of implants and they actually technically get pregnant. And we think that there's a couple months between those, and that maybe like the success of the breeding and the success of the implantation is what ultimately drives pregnancy, and that the success during the foraging trip is what allows them to keep their pregnancy. So there's these sort of like checkpoints, and we're trying to work with, physiologists and folks. A lot of the people in Dan Crocker's lab at Sonoma State and Dan Costa's lab here are trying to figure out exactly, you know, how they implant, when they implant, why they fail, what the drivers are. But the short answer is an anecdote, which my former undergrad student and now tech, Natalie Storm, did for her senior thesis, which is she looked at what we call early arriving females. So there's this subset of females that comes to the beach, they're reproductive age, but they don't show normal reproductive timing. So instead of coming to the breeding beach at that normal time in January, February, they actually haul out with the juveniles, like right now in November-ish. And then they skip the entire breeding season. They come back for the molt, then they come back the next breeding season and have a pup. So somehow they got pregnant, but they weren't here for the breeding season when the big males were here. And they surely weren't inseminated by the males in either the molt or the juvenile haul out. And so the question is, when are these females getting pregnant? Like, they have to be inseminated for them to get pregnant. That's just how that works, right? So what's happening there? And the answer comes a little bit from the southern elephant seal literature, in which one researcher tagged one adult female with an instrument on her head, and she went out for her long foraging trip. And she came almost all the way back, like thousands of kilometers. But before she got to land, she turned back out and went back to sea, and then hauled out for the next breeding season and had a pup. And so the only way that she could have gotten pregnant is by breeding in the water. There's no land around. She didn't haul out on some island somewhere. So clearly they breed in the water. We've actually seen it off the Channel Islands a couple times. There's some photos. There's one in a book chapter that I published a couple years ago. But we've never seen that. Like, northern elephant seals don't really breed off Año Nuevo in the water. So we've never seen that. But clearly, if they're not breeding on land and they're coming back with a pup, they had to breed somewhere. And we think that breeding in the water is probably the only way that can happen. But again, like, one of these many unknowns that will hopefully resolve in the next 20 years or so.
I have a quick follow-up question. I know earlier you showed a slide that showed a 20-year-old female who had a pup in her 20th year. So do they not, can they reproduce to the end of their lives, or is there like a certain time frame?
Yeah, that's a super good question. It's actually one that I showed you guys the example of the class project looking at the oceanography. The class the year after that, this past cohort, decided to look at reproductive senescence, and they were looking at 7 different ways that elephant seals could senesce. So like, in humans, we experience menopause, we can't reproduce to the end of life. So our lifespan far outlives what we call our reproductive lifespan. And then we have that like menopause period. And the existence of that period in wild animals is pretty hotly debated. Like, it's been found in like, I think 3 total species, like a couple cetaceans and I think like a shrew or something. And so we wanted to see, what that looks like in elephant seals. And it looks like they can reproduce up until their last years, but their reproductive success, like the proportion of successful pups, drops off at the end of life. They tend to produce a lot more males at the end of their life. Their pups tend to be a little bit less successful in both surviving and reproducing themselves. And so that's a paper that Allison Payne is now sort of leading to wrap up to submit to the Journal of Animal Ecology. But it's this really cool story that elephant seals do experience some sort of senescence at the end of their lives that manifests in lower reproductive success and slightly lower quality pups, and definitely skews the sex ratios of the pups that are being born. So it seems like maternal age does have a really important impact on how the seal pups are able to be successful.
Thank you.
Paul asks, what are your thoughts on how the elephant seals navigate back to the same place every year?
That is the biggest question in the navigation literature, and one that no one has been able to answer in any species so far. So what are the actual cues behind navigation? Some people think it's stars, like, oh, they know the stars and they can align. Some people think it's some sort of sound, like listening to waves break on the shore. Some people think they use social cues. Some people think they use a magnetic sense. And maybe the diving of elephant seals is adaptive, not just for eating, but also for getting closer to the earth, and therefore the magnetic field being stronger to be able to pick up cues. It's been a whole thing, and people have tried from an anatomy perspective, from a behavior perspective, from a modeling perspective, to try to disentangle all of these cues. And my perspective is that, like everything in science, there's probably not a single cue that they're using. Like, they've evolved to have backups for everything, right? And so sure, they probably use some magnetic cues, and they probably use stars when they can, and they probably have some sort of like timing mechanism in their brains. But it's probably a combination of all those things that basically allows them to get back on time. And how they actually do that, I'm not sure we'll ever know, but it's a pretty interesting question, and one that I'm hoping as we deploy more and more of these instruments and get a better and better sense for sort of weird things that happen, like, when a solar eclipse happens, like what do the seals do? When there's a magnetic anomaly in space, like not in outer space, but like in spatial locations, then like what do the seals do? We can chip away at those questions, but it's really like a huge question that I'm not sure will ever be answered in wild animals.
Paula asks, is the elephant seal population at a healthy number at this time? I'm wondering how the warmer water temperatures are affecting them.
Yeah, so healthy number, it's a loaded question. Elephant seal populations at Año Nuevo seem to have sort of plateaued. We think they're at what we call carrying capacity, which means there's like some resource in the universe, either like prey or like space on the beach or something, that's basically limiting the number of seals that can exist in the wild. Whether that's true for all the populations, we don't know. You can really only answer that question if you're doing censuses of other populations, or if you're, during mark recapture, you can see how their survival and reproductive rates and like pup size changes over time. The seals have reached huge numbers. It's probably something like, I don't know, 240 or 250,000 now. It's a lot of seals. So they seem to be doing well. I will say it's a little bit stressful to watch the, avian flu situation develop in the Southern Hemisphere. There's been a lot of mass mortalities in seabird colonies and then in seals that are nearby seabird colonies. And there's a lot of fear, I think, that the elephant seals are so genetically similar to each other. Millie, my student, likes to say they're little clones because there was such a bottleneck, and now, the population is rebounded, but the genetic distinction between individuals isn't that high, and that makes them potentially really susceptible to these pathogens that come and sweep through entire populations. And so while their numbers seem to be doing really well right now, they may be affected by things like pathogens that come in unexpectedly from places and could potentially have a pretty devastating impact on the population. It seems like over the last few years, the numbers of pups being born have dropped like a little bit. And we're not sure if that's oceanography. It's been a pretty tough couple years in terms of a couple of the oceanographic indices like aligning in a bad way, where there's like a bad El Nino along with a bad heat wave kind of thing at the same time. And that has these little bumps in the population. But in the grand scheme of things, I think elephant seals are doing pretty well, and we're hoping it's going to stay that way for a long time.
So Stuart observes that you don't have an entrance gate at Año Nuevo from the sea. And he wonders how then you reduce errors with regard to observations of when they arrive.
Stuart, after my own heart. Año Nuevo has an entrance gate. It's manned by the state park, and the elephant seals choose to use the giant back door, which is not very helpful for us figuring out when they arrive. In short, we have special computer algorithms that we use to figure out when the seals arrive and depart. You must be a very astute person, because I had originally, in the paper I just submitted to Proceedings B, I had used first observation and last observation as my metrics, like when was the first time we saw them and the last time we saw them. And one of my co-authors said, there's a lot of error associated with these dates, because we don't always see the animals every single day that they're there. The detection probability is something like 60 to 90%, depending on the person who's doing the observations and how experienced they are. And what's worse is that the dates are biased, right? You can never see a seal before it arrives, but you may miss it after it arrives, and vice versa. When it's departing, you can never see it after it's departed, but you can miss it if it hasn't departed yet. And so we wrote this algorithm to basically correct for that. And the spoiler alert is we were off by, on average, 0.1 days from the seal's actual arrival dates on average. So we're pretty good at our jobs, but you're right, we're not perfect. So we have to use those algorithms to make those corrections. And now the paper has departure and arrival dates instead of first and last observation, which took about a week of my time, but it's totally worth it for the quality of the science.
Paula asks, what causes the juvenile males to return to the beach in October?
Yeah, I don't know. Both the males and the females come to shore in the juvenile haul out in this time of year. There's been a couple hypotheses about why that happens. Personally, my sort of like spidey sense is that the juveniles just want nothing to do with the breeding season. They're like, it is chaos, it is dangerous, it is cold. And so they don't need to come for the breeding season. They don't need to do any like social learning, and they don't need to do any breeding. So they're just kind of just trying to get through and survive until they are sexually mature and can therefore show up to the breeding season and make a difference. Some of the other like slightly crazier hypotheses are like, someone told me once, I think it's to develop their bone density. It's like, how astronauts have to like run on treadmills in space so they don't lose their bone density. It's kind of the same idea. Like, imagine being an elephant seal and just like floating in like endless buoyancy for like 8 months at a time, and then coming to land and all of a sudden being 500 kilograms and having, or like in the case of males, 2,000 kilograms, and having all of that weight be sitting on you. So maybe they come back to sort of like experience that and builds their morphology somehow. I don't know. I think that's a pretty funny hypothesis, but maybe it's totally true. So there's a couple potential hypotheses, but no one's really ever studied it. Just a couple of years ago, someone for the first time actually quantified how long that haul out period is, which is about 35 days. So we're really in the beginning stages of understanding juveniles, and we have a lot of work to figure out like why it is that they do what they do and how successful their strategies are, but we're not quite there yet.
So Anthony asks a question. You may have answered this, but maybe you'll have a new insight to share. So do individuals move between colonies, such as between Año Nuevo and Piedras Blancas? Maybe within a given year, do they ever move between colonies?
Yeah, they definitely do. I will say that the movement is more one direction than the other. So we see a lot of white tags at Año Nuevo. That's from Piedras Blancas down south. And Piedras Blancas sees some green tags, but not a ton. And so it would take a mathematician to know, is that random chance or not? If you were paying attention, there's like 10 times as many seals down south as there are north. So you would expect more seals to be coming north than you would south. But the numbers seem pretty imbalanced to me. And so it wouldn't be surprising to me if there was a lot more movement north. And I think that supports this sort of like spatial recolonization idea. And also the idea that the bigger colonies are really getting overloaded with seals and they're trying to look for somewhere else to go. So there's not a ton of research right now happening on this sort of movement between colonies, but it's something a lot of us are thinking about. And it'd be really fun to do a big paper across all the colonies to see if we can pool all of our observation data and calculate some better numbers, because right now our numbers are pretty rough.
You may have addressed this a little earlier, but Joanne asked, can you address how long it takes a female to get to beach to birth and how she knows to get back or when she will be in labor?
Yeah. I don't know the answer to the second question, how seals know when they're supposed to be back. I can tell you how long it takes them to get back, and it is extremely variable. So like, even though they're genetically really similar, the seals are what we call phenotypically, or like trait based, very different across individuals. So they go to really different places. Some go 1,000 kilometers, some go 5,000 kilometers maximum distance from Año Nuevo. And so the MapSense paper found that, if the seals are foraging 5,000 kilometers away, they're turning around and heading back to the beach a lot earlier to be able to give themselves the time to get back. We found that they have this like maximum cruising speed at something like 120 kilometers per day. That's really far. But they can't speed up past their, it's like their biomechanics and their body mass and their transport modality don't let them go any faster. So we do see that they modulate their speed. If they get behind, they do actually speed up, but there is that maximum cruising speed and they can't really go faster than that. So how long does it take them to get back? Well, the answer is however long it takes, depending on how far they are and how much they can modulate their speed.
And will they always get back in time to be in labor, or is there like a miscalculation sometimes?
It's a really good question. The short answer is I don't know of any examples of them not getting back in time, but if they didn't get back in time, I probably wouldn't have that answer. Because what they would do probably is give birth at sea and then come onto land and not have a pup. And we would just observe them day after day after day after day. But instead of at the 5-day mark producing a pup, we would see them at day 6 and day 7, day 8, day 9, day 10 without a pup. We see that sometimes, but as you can see from this video that has stopped playing, the breeding season is just chaos. It's like which pup is which. Like you can see, okay, this pup belongs to that female, this pup belongs to that female, this pup belongs to that female. Like, is that a female? Looks like a female. Where's her pup? I don't know. This one in the front is flipping sand and looks really uncomfortable. If you like watch that female, you can see, I think she's actually pregnant and is, you can actually see her contractions on her side. She's like, oh God, what's happening? I'm going to flip sand. I think I'm going to have a pup now. And so it's just chaos. So we stand there, we watch this, and we can just watch like 2 females will swap pups and not even notice. And so one of the questions that my student Millie has for her dissertation is how often are pups that we think belong to known moms, were they actually born to those moms? So we can take genetic samples from the mom and the pups. We can use the observations from the mom and the pups and compare those 2 answers and see how different they are. And therefore how frequently the observational data are wrong because we observe a pup with a mom that's not actually like the pup's genetic mom. Does that make sense? So it's a little bit hard to tell if we see a female without a pup, whether she never had a pup in the first place or whether her pup was a stillborn or didn't make it or, you know, was born at sea. But we had a reviewer of that paper say, well, what if it's hauling out that makes them give birth? Like, what if it's the gravity associated with being on land that actually like causes them to go into labor? And it's like a chicken or egg thing. Like, I don't know how to even begin to do the science behind whether gravity is what induces labor in elephant seals. But I think it's a really good question of like, is it being on land that induces pregnancy, or is it that they get back right in time to give birth to that pup? And I don't think we'll ever know the answer to that question.
Thank you.
So Becky wonders about the southern elephant seals versus the northern elephant seals, and do they get together? Do they interact? And maybe is there a genetic difference?
Yeah, that's a great question. We included one southern elephant seal genetic sample in our project with Millie, and it's pretty different. If you, I wish I could do a poll, but if you've ever seen a southern elephant seal, you know that they are very different looking than northern elephant seals. The males have much smaller noses. They're much bigger. The females have like huge, broad faces. They're a lot uglier than the northern elephant seals are, but don't tell the southern elephant seal researchers that. And we're constantly trying to figure out like what is similar and what is different. I actually, I'm really hoping one of the grad students who starts in my lab next year might be interested in doing some comparisons between northern and southern elephant seals, because there's a lot of really interesting differences in their oceanography. There's differences in their mortality rates. There's differences in how often they give birth. There's differences in their pup size. And if you compare the demography of 2 species using the same methods, you can begin to understand the differences in their environments. Like, are there more predators in the Southern Ocean than the North Pacific? Is there more prey in the North Pacific than the Southern Ocean? And I can tell you from some of the work that Joffrey's done that the diving behavior is pretty different in juveniles. We see a lot more predator avoidance behavior in Northerns than Southerns. And so that sort of signals to us that there's probably differences in the composition of the ecosystems and sort of the ecology that goes from the bottom to the top, from oceanography all the way to top predators. And we have a lot more to learn about that. But the short answer is northern and southern elephant seals don't actually mix in their current geographic distributions. But there are some wandering seals. There's some evidence, some published evidence of southern elephant seals being found in like, I think, really, really southern Mexico. So it's not impossible, but we don't really see it yet. They're really, really far south.
Got it. Thank you. David, do you want to do 1 or 2 more questions, or?
All right, most one. Why don't you go for one more? And I think that has to be it.
Okay. So Stuart asks, what percentage of the pups die on the beach due to being trampled or with diseases or predators or something else?
Yeah. It's an interesting question because I'm super interested in mortality, as you can probably tell. I think it's super interesting to figure out how wild animals die, because in most species we don't know. And as adults, we don't see dead elephant seals. Like if they die, they basically die at sea. And so it's the lack of their presence on the beach that signals to us that they died. Or in the case of satellite tagged seals, which is a really small subset, we can actually like watch those go offline. And because we know we affix the instruments really, really carefully and really strongly, if a seal stops transmitting, we know that that seal is probably no longer alive. So we can get like little bits and pieces indirectly of how the adults are dying, but the pups, they actually die on land during the breeding season, like you're asking. And that's a really interesting case study, because we can actually count how many dead pups are there, how old were the pups when they died. Like you can see in this video, you can actually tell how old pups are by how big they are. Like, this one here is probably like a, I don't know, like 6 or 7 day old pup. This one here is like a 2 or 3 day old pup. It's pretty small. If you look in the back here, you can see some like really, really big pups. And so we can tell how far along they were. And then of course, the people who, have experience with necropsies can go and do the necropsy and figure out cause of death for the pups. And the short answer is it's a pretty substantial amount of pups that die. I don't know the exact numbers. Like if I had to guess, maybe 5% of pups or something like that. But it's super dependent on things like the year and the males that are around. Last year, we had 2 crazy things happen around the time that I gave a crawl lecture. I think it was January 2022. There was a tsunami, and basically like the tsunami came at the same time as the tide was like a king high tide, and the swell was huge. And it was just this like awful combination in January. So when like most of the pups are being born, of like all these factors coming together at the exact wrong time. And like somehow it was like almost totally fine. Like we watched a couple of pups get washed away, which was super sad. But for the most part, the pups and the moms actually sort of like moved up against the cliff, and like the moms like put themselves on the ocean side of the pups to sort of like protect them. And it worked. It was really successful. We've seen other times when, for example, there's this like new, as you know, the climate is changing the beaches a lot in California. There's a lot of like coastal erosion, and the dunes move a lot. So what's a beach in one year can be like a dune in the next. It changes really quickly. And there's this new gully that was created the last couple of years, and females started breeding in it. And it's this gully that has like, it's kind of a bowl shape. And what that means is that a lot of the pups and the females are in the bottom of the bowl. And there's only, like talking about entry gates, there's only one in.
Oh.
I'm back. No idea.
Oh, that was weird.
Yeah. Apparently Skype reset, or Zoom. Oh, I'm in Skype land. Anyways, in that particular gully, there was a lot of mortality for the pups because the males were basically trampling all the pups in that one gully. All the pups would like roll down because they're all like fat and roll. And then the males would just come through and try to get through during a big fight and just smash all the pups. So I think we saw like 25 or 27 dead pups all in this one little gully. And that was a weird anomaly thing. But we'll be curious to see this year, like, are the females going to breed in that gully? Do they learn? Did they watch that happen? Do those females that bred in that gully go to different beaches? Like, is their sort of micro habitat fidelity a lot different after they've experienced that sort of catastrophic pup loss? So these are the types of questions that we can answer using these really cool long-term data sets. And it's one of the reasons that we're super excited to go out to Año Nuevo, even if it's cold and wet and rainy and early and we're tired, to be able to collect the data to answer all these exciting questions and to like set up the next generation of researchers to do the same. So we're feeling really lucky.
Amazing.
And marvelous. Well, wow, thank you very much. Please join me in applause for Assistant Professor Roxanne Beltran. You can put your comments down in the Q&A box. And thank you, Professor, for sharing your fabulous research and your researchers with us this evening. Now, keep in mind that a couple of links have been given down in the chat box. If you want to support the Beltran Lab, follow that link. If you want to join the professor as she goes off to the Galapagos, follow that link. We certainly encourage giving a gift to those cold researchers out there. Also want to thank the staff of the Alumni Relations and Special Events Offices who organized this webinar. Thank you, Shana, Diana, Paulina, Nicole, and Kristen. Also Mike Reapy, the co-organizer of the series. Well, our next Slugs and Steins will be Monday evening, December 11th, with Assistant Professor Nia Jones, with the talk title, Teaching Beyond Crisis, Black Ecologies and Critical Hope. Dr. Jones is an assistant professor of sociology and core faculty in the Global and Community Health Program of UCSC. In the Slugs and Signs talk, Dr. Jones will reflect on teaching Black ecologies amid ongoing social and environmental injustice, including pandemic inequities and climate change. Her current projects broadly focus on the study, teaching, and practice of Black environmental knowledge in the 21st century. Her award-winning solo and collaborative projects have been supported by the Robert Wood Johnson Foundation, the Botanical Society of America, and the American Association of Geographers. Well, meanwhile, UCSC has other exciting events awaiting. This Wednesday, for example, November 15th, will feature Professor Emeritus of Theater Arts Paul Whitworth, who will advise us on how Shakespeare's King Lear provides a cautionary tale for retirees. The next day, Thursday the 16th, punk rock and its role in social change will be discussed while celebrating the launch of 2 new books. And on December 6th, both in Santa Clara and online, the Crawl Lecture will address bridging the gap between artificial intelligence and natural intelligence with Jason Eshragian of UCSC's Department of Electrical and Computer Engineering. More on these events and others at events.ucsc.edu. So, on behalf of the UC Santa Cruz Alumni Association, thank you so much for joining us. Please come back on December 11th at 6:30 p.m. for our next virtual event.
Thank you. Bye, everyone. Good night.
Good night.